Hi everyone. You're watching the Top Wrench channel, and I'm John Capello. I'm completing an Anatomy and Physiology project here, which is the reading of Principles of Anatomy and Physiology, 12th edition. I had originally had a thought that I would narrate for Perkins School of the Blind. There were a lot of applications, and I got lazy and I just didn't do it. That's a fragile community, and so there are all kinds of things that you have to do and things that you have to pass, and I said, oh wow, paperwork. I'm not going to do that, so I didn't do it. But now I figured, I'm going to read this. It's a book I've read anyway, not cover to cover. I did take the class, but I think that reading it cover to cover or hearing it cover to cover is just one way that you can hear the material the first time, or the second time, or the third time, because like I've stated earlier, as far as I'm concerned, I don't know, I'm sure there's wizards out there that can absorb material as fast as they read it, but I feel as though I took A&P 1, I took A&P 2, I could take A&P 3 more times and probably come up with something new every time I take it. It's a very in-depth subject. So right now, I'm finishing up Chapter 3, and it's meant for that audience. It's not meant for its video production quality. Although, to delve into those waters, that might be great. And I'm not saying I'm not going to do that later on. That would be awesome. I would love to be able to do that. But right now, this is about narration and getting the material into a form that's usable. Not about my graphics or my ability to manipulate that. But that is a great talent. I'll get into that later, I hope. So right now, we're on hypertonic solution. And I gotta say, I've recorded this same section 5 times, and I end up swearing halfway through it because I don't want to edit. I want to just move right through it. And if I miss a word, that's okay. We'll go back, and I'm trying to do it in such a way that if I did want to edit it, the section without error is in there. It might be right next to the section that I made a mistake. I mispronounced a word, and I'll say, okay, let me repeat that, and I repeat it. And so if I did want to go back and cut that out in the audio, I could have it. But I don't think it's really important. If somebody misses a word, if your teacher is up there and she misses a word or he misses a word, you don't miss the whole subject. You don't miss the whole point of it. You say, oh, okay, you arrive at it eventually, and that's what I want to do. I want to do it as a lecture, as a reading. But just try to stick to the data. So I'm going to continue with the hypertonic solution. And this is part of Chapter 3. And a hypertonic solution has a higher concentration of solutes than does the systole inside red blood cells. One example of a hypertonic solution is a 2% NaCl solution. In such a solution, water molecules move out of the cells faster than entering, causing the cell to shrink. Such shrinkage of cells is called crenation. The clinical connection, medical uses of isotonic, hypertonic, and hypotonic solutions. Red blood cells and other blood cells may be damaged or destroyed if exposed to hypertonic or hypotonic solutions. For this reason, most intravenous solutions, liquids infused into the blood of a vein, are isotonic. Examples are isotonic saline, 0.9% NaCl, and D5W, which stands for dextrose 5% in water. Sometimes infusion of a hypertonic solution such as mannitol is useful to treat patients who have cerebral edema, excess interstitial fluid in the brain. Infusion of such a solution relieves fluid overload by causing osmosis of water from interstitial fluid into the blood. The kidneys then excrete the excess water from the blood into the urine. Hypotonic solutions, given either orally or through an IV, can be used to treat people who are dehydrated. The water in the hypotonic solution moves from blood into interstitial fluid and then into body cells to rehydrate them. Water in most sports drinks that you consume to rehydrate after a workout are hypotonic relative to your body cells. Active processes, active transport. Some polar or charged solutes that must enter or leave body cells cannot cross the plasma membrane through any form of passive transport because they would need to move uphill against their concentration gradients. Such solutes may be able to cross the membrane by a process called active transport. Active transport is considered an active process because energy is required for carrier proteins to move solutes across the membrane against a concentration gradient. Two sources of cellular energy can be used to drive active transport. Energy obtained from hydrolysis of adenosine triphosphate is the source in primary active transport. Energy stored in an ionic concentration gradient is the source in secondary active transport, like carrier-mediated facilitated diffusion. Active transport processes exhibit a transport maximum and saturation. Solutes actively transported across the plasma membrane include several ions such as sodium, potassium, hydrogen, calcium, iodine, and chlorine. Amino acids and monosaccharides. Note that some of these substances also cross the membrane via facilitated diffusion when the proper channel proteins or carriers are present. Primary active transport. In primary active transport, energy derives from hydrolysis of ATP changes the shape of a carrier protein, which pumps a substance across a plasma membrane against its concentration gradient. Indeed, carrier proteins that mediate primary active transport are often called pumps. A typical body cell expands about 40%. A typical body cell expends about 40% of the ATP it generates on primary active transport. Chemicals that turn off ATP production, for example, the poison cyanide, are lethal because they shut down active transport in cells throughout the body. The most prevalent primary active transport mechanism expels sodium ions from cells and brings potassium ions in. Because of the specific ions it moves, this carrier is called the sodium-potassium pump. Because a part of the sodium-potassium pump acts as an ATPase, an enzyme that hydrolyzes ATP, another name for this pump is sodium-potassium ATPase. ATP with the letters ASE afterwards, ATPase. All cells have thousands of sodium-potassium pumps in their plasma membranes. These sodium-potassium pumps maintain a low concentration of sodium in the systole by pumping them into the extracellular fluid against the sodium concentration gradient. At the same time, the pumps move potassium into the cells against the potassium concentration gradient. Because potassium and sodium slowly leak back across the plasma membrane down their electrochemical gradients through passive transport or secondary active transport, the sodium-potassium pumps must work non-stop to maintain a low concentration of sodium and a high concentration of potassium in the systole. The different concentrations of sodium and potassium in systole in extracellular fluid are crucial for maintaining normal cell volume and the ability of some cells to generate electrical signals such as action potential. Now, I'm going to pause here for a minute in the book. I'm going to read that to you again. The different concentrations of sodium and potassium in systole in extracellular fluid are crucial for maintaining normal cellular volume and for the ability of some cells to generate electrical signals such as action potential. Action potential, as you will find out when we cover the nerve channel, is very important. Action potential is, as it says, the potential to create action. That action is created electrically because, as you know, our bodies run on electricity. Not the kind of much like the kind of electricity that is in your wall socket, in your flashlight, but at a minute level. The voltage of the cell is 200 millivolts. Now, in the action potential that we're discussing is so minute that two positive ions, sodium and potassium, are used, the trigger being calcium. My point being here is that the action potential, or the minute difference in charge, is what causes the flow of electrons, the flow of ions. Excuse me, I meant the flow of ions. Whereas the difference in two substances, for instance, nickel and cadmium, that provides the action potential of your battery. It really is no different. And so that's something to think about, is how very similar all of these disciplines are. Whether it be anatomy and physiology, electrical engineering, plumbing. It's all one thing. It's all one thing, but becoming more and more in depth depending upon what your discipline of choice is. And you may find that based upon your discipline of choice, you can get to all of these other disciplines. And there isn't much of a difference other than the details. So I'm going to begin that paragraph again and continue. The different concentrations of sodium and potassium in systole and extracellular fluid are crucial for maintaining normal cell volume and for the ability of some cells to generate electrical signals, such as action potential. Recall that the tonicity of a solution is proportional to the concentration of its solute particles that cannot penetrate the membrane. Because sodium ions that diffuse into a cell or enter through secondary active transport are immediately pumped out, it is as if they never entered. In effect, sodium ions behave as if they cannot penetrate the membrane. Thus, sodium ions are an important contributor to the tonicity of the Thus, sodium ions are an important contributor to the tonicity of the extracellular fluid. A similar condition holds for potassium in the systole. By helping to maintain normal tonicity on each side of the plasma membrane, the sodium-potassium pumps ensure that cells neither shrink nor swell due to the movement of water by osmosis out of or into the cell. The secondary active transport. In secondary active transport, the energy stored in a sodium or hydrogen concentration gradient is used to drive other substances across the membrane against their own concentration gradients. Because sodium or hydrogen gradient is established by primary active transport, secondary active transport indirectly uses energy obtained from the hydrolysis of ATP. Now, once again, I'm going to go back and deviate from the text for just one more hypothesis, which is mine. People have always wondered, why do squirrels run when it rains? How do they know that it's going to rain before it rains? And this is a mystery to them. It's not a mystery. Now, we're under constant adjustment, as per that first paragraph that I just read. It says, by helping, I'm going to backtrack here, by helping to maintain normal tonicity on each side of the plasma membrane, inside the cell and outside the cell, on each side of the plasma membrane, the sodium-potassium pumps ensure that cells neither shrink nor swell due to the movement of water by osmosis out of or into the cells. Well, that may happen, as it states, many times a second, thousands of times a second. However, it would seem that that would be a feedback loop. It would begin to shrink or it would begin to swell, meaning the cell itself, before the correction can take place. You can't pump out sodium that's not in there. You can't pump in sodium that's not outside. It has to be corrected. It's a correction that's made. Now, in the instance of our weather, you hear constantly barometric pressure. Well, what is that? There are experiments right in this book, I believe it's in this book, or it could be in the chem book on micro, in which they take a balloon, and the balloon is put inside of a vacuum chamber. Now, when I say vacuum chamber, I simply mean a chamber that is sealed to the air, the outside air, the ambient air, and it has a vacuum tube on top of it so that you can apply vacuum. Inside of the container, like this, if this were upside down, this jar, and there were a lid on it, but on the inside of the lid was a balloon. Just a balloon that was sealed to the inside of the lid, and then the lid was screwed on, and then, now I would say that this would be upside down, that, it would be inverted, and there were a vacuum tube completely sealed to the ambient air on the top of it. In such a setup, if you applied vacuum to the vacuum chamber, you would find that the balloon expanded and blew up. If you remove the vacuum, the balloon would deflate. That is much like our atmosphere. When we have a low ceiling, when we have clouds, barometric pressure is low. I believe that your cells are like little balloons in your body, little cells. When that vacuum pressure, when that barometric pressure lowers, you can experience a minute expansion of your cells that needs to be created. A lot of these cells, your muscle cells, your nerve cells, you can feel. Of course you can feel your nerve cells. And so this is probably the root of fibromyalgia, of arthritis, why people with arthritis, why people with migraine headaches, why we suffer when the weather changes. The weather changes, barometric pressure reduces before a storm. You'll get an expansion of the cell, which is corrected. Perhaps there's a lag. There has to be a lag in a feedback system. Because without a problem, you can't fix the problem. So there has to be a problem for the feedback to work. So this pressure on these tiny little animals that scurry about, your squirrels, your chipmunks, your mice, they're freaking out because all of a sudden it feels like death. It feels like there is a danger approaching. And to a little chipmunk, a storm is an absolute danger, because when that chipmunk's hole gets flooded, you got no more chipmunk. So I think that's the reason behind why they scurry around and why we get achy joints and headaches. And so it says that by helping to maintain normal tonicity of each side by helping to maintain normal tonicity, a normal solution, tonic solution, on each side of the plasma membrane, inside and outside the cell, the sodium-potassium pumps ensure that cells neither shrink nor swell due to the movement of water by osmosis out of or into the cell. So think about that. Now, I'm going to repeat this paragraph again. Secondary active transport. In secondary active transport, the energy stored in sodium or hydrogen concentration gradient is used to drive other substances across the membrane against their own concentration in gradients. Not ingredients, gradients. Once again. In secondary active transport, the energy stored in a sodium or hydrogen concentration gradient is used to drive other substances across the membrane against their own concentration gradients. Because a sodium or hydrogen gradient is established by primary active transport, secondary active transport indirectly uses energy obtained from the hydrolysis of ATP, adenosine triphosphate. The sodium pump maintains a steep concentration gradient of sodium across the plasma membrane. As a result, the sodium ions have stored or potential energy, just like water behind a dam. That reads like a typo because it uses the word or. As a result, the sodium ions have stored or potential energy, just like water behind a dam. Once again, the sodium-potassium pump maintains a steep concentration gradient of sodium across the plasma membrane. As a result, the sodium ions have stored or potential energy, just like water behind a dam. I think that should read, as a result, the sodium ions have stored potential energy, just like the water behind a dam. Accordingly, if there is a route for sodium to leak back in, some of the stored energy can be converted to kinetic energy, energy of motion, and used to transport other substances against their concentration gradients. In essence, Secondary active transport proteins harness the energy in the sodium concentration gradient by providing routes for sodium to leak into cells. In secondary active transport, a carrier protein simultaneously binds to sodium and another substance and then changes its shape so that both substances cross the membrane at the same time. If these transporters move two substances in the same direction, they're called symporters. Antiporters, in contrast, move two substances in opposite directions across the membrane. Plasma membranes contain several antiporters and symporters that are powered by the sodium gradient. For instance, the concentration of calcium ions is low in the systole because sodium, calcium, antiporters eject calcium ions. Likewise, sodium-hydrogen antiporters help regulate the systole's pH by expelling excess hydrogen. By contrast, dietary glucose and amino acids are absorbed into cells that line the small intestines by sodium glucose and sodium amino acid transporters.